In further detail, RNA is the template in protein synthesis, so Nierenberg and Matthaei published their findings that aimed at proving the RNA relationship, but it also revealed some clues into cracking the genetic code. However, the two created a synthetic RNA that did not require a template like other RNA polymerase. This new synthetic RNA was created by the enzyme polynucleotide phosphorylase and was found to stimulate protein synthesis. The synthetic RNA only consisted of the base uracil, codon UUU. So the hypothesis was that a triplet codon based on 3 base codons translates into specific amino acids. E. coli was selected as a source of cytoplasm. The E. coli extract was added to 20 different test tubes, and the extract contained tRNA, ribosomes, amino acids, and tRNA synthase, which is just a charged tRNA. The synthetic mRNA comprised of uracil nucleotides and only UUU codons was added to all 20 test tubes too, also known as poly-U. The results were that in one test tube a protein was made. The amino acid phenylalanine Phe bonded repeatedly to itself, also known as a homopolymer. Homopolymers are polymers composed of only one repeating unit in their molecules. The polypeptide produced contains a single amino acid. From the results, Nierenberg and Matthaei proceeded to repeat this experiment, however using different synthetic mRNAs comprised of cytosine and then adenine. As the same rules of mRNA apply, it only contains one repeating base. These two other bases produced results that CCC encoded for proline and AAA encoded for lysine. You can also see here the mRNA strands for the different codons, and then you can see the amino acids that it translated into. Now on to Nierenberg and Letter's experiment. Random copolymers were used to try and determine the remaining amino acid codons, however nothing was revealed. So a couple years later, Nierenberg and Letter took advantage of the new technology that allowed for all codons, not just homopolymers, to be made in vitro. They developed a new technology that took small sequences of mRNA and realized that even then it would bind to a ribosome. They first made artificial short mRNA codons and mixed ribosomes and tRNAs, as you can see here. Secondly, they pass the mixture through a filter that traps the ribosomes at the top and lets the tRNAs not bound to the ribosome pass through the filter. 50 short mRNAs with known codons were synthesized and added individually to mixtures of ribosomes and tRNAs with amino acids. The tRNAs that were bound to the mRNAs and ribosomes were then isolated from there where they were able to determine which amino acids were present. Through these experiments, the entire genetic code was cracked, and as you can see here, this is the chart that they discovered.